As you remember, neurons communicate with one another by means of electrical signals within a neuron, which then send a chemical signal across the synapse to downstream neurons. We can eavesdrop on these electrical signals by sticking an electrode into the brain and measuring changes in voltage over time. One way to do this is to stick a very small, sharp electrode tip right through the cell membrane and into a neuron. This is called an intracellular recording. You can also just get the tip of that electrode close to the neuron on the outside to perform what's called an extracellular recording. Extracellular recordings are a little more robust to small movements of the tissue relative to the electrode. So they're more typically used when you're recording from an animal that's awake and moving around. Here's a plot of what the voltage over time looks like when it's recorded with an extracellular electrode. Notice that the magnitude and shape of the action potential that you record in this way is a little different than what you see with an intracellular recording, but the time course is about the same, lasting just a few milliseconds. When we're looking at the activity of a neuron, we rarely care about zooming in on individual action potentials, which tend to be the same every time. Instead, we care about the firing rate of the neuron over time. Each action potential, or spike, is assigned a 1 millisecond bin, and we look at how many action potentials occur over some slightly larger bin, say 25 milliseconds. We can then plot this firing rate over time. We align the firing rate of the neuron to some external event, for example, the time when a visual stimulus appeared on the screen. And usually the firing rate we plot in a PSTH is the average, obtained over multiple presentations of the same stimulus to account for variability in neural responses from trial to trial. Often, PSTHs for two different conditions will be plotted side by side, or even on the same axis for easier comparison. Each line reflects an average over many trials of the same type, aligned to the same events, such as stimulus onset. In this example, you can see that a neuron in V1 has a strong visual response to a vertical bar, but a much weaker response to a horizontal bar. Of course, we can only perform these sort of invasive electrophysiological recordings in animals, not in human volunteers. In the next video, we'll talk about a way to measure human brain activity.